We usually think of DNA mutations as just random errors, right? Often harmful things our cells are, like working nonstop to prevent.
Exactly. Our bodies have this incredibly faithful machinery. It fixes thousands of DNA glitches daily.
Yeah, and makes unbelievably few copying errors, like super accurate.
hmm But what if we told you that sometimes living things intentionally rewrite their own genetic code?
Intensely. Like they program changes to their DNA.
That's the idea. It's a fascinating twist on how we usually view genetics. Imagine having this perfect blueprint, but then realizing certain parts are deliberately altered for specific purposes.
Exactly. So today we're doing a deep dive into something called endogenous genome editing.
Right. And this isn't CRISPR in a lab, though, it shares some ideas.
No, this is nature's own thing. Built-in systems for making really precise, targeted changes to DNA as part of, well, normal life.
We're going to explore how organisms manage these genetic feats and when these pretty incredible abilities come into play, especially in really critical things like, say, immune defense.
Our mission today is really to unpack this. It's complex, but it's also incredibly impactful biologically.
Yeah, and you'll discover how editing systems that look totally different across the tree of life often rely on surprisingly similar genetic tricks, regardless of the specific job or the organism.
Get ready for some serious aha moments, hopefully. It
Uh,
might change how you think about genetics.
Definitely.
Okay, so let's start with this core paradox. DNA is usually fiercely protected.
Like you said, human cells repair thousands of damaged sites every single day. Replication is incredibly accurate, maybe one error per billion bases copied.
Just amazing fidelity.
Yeah.
Yet, in stark contrast, some organisms programmably induce targeted DNA changes. They alter their own sequences on purpose.
And what's truly astonishing is that this isn't some rare fluke. This endogenous genome editing, let's call it EGE, it's actually widespread.
Really? Like where?
We see it in all vertebrates, at least half of all prokaryotes, so bacteria and archaea and lots of other types of organisms too. And the scale varies wildly. It can happen millions of times per generation in some species, or maybe just once every 10 million generations in others. But the underlying molecular strategies often share a common playbook.
And it's not a new discovery either, is it? I read the first hints were like over a century ago.
That's right, 1887, in a nematode worm, Akker S. And even if EGE only alters a tiny fraction of the genome, those changes can be disproportionately important for how the organism functions.
Right. Think about it. This natural ability has even been the basis for huge biotechnologies like CRISPR-Cas9 and also antibody engineering.
So just to be really clear on our terms, EGE is an organism making programmed changes to DNA sequences at specific targeted locations within its own genome.
Okay, and it's important to understand what it isn't.
Crucial, it's not those random, spontaneous changes like most single nucleotide variants or the kind of hypermutation you see in cancer. That's different.
Right, and it's not global, untargeted stuff like the big gene shuffling during meiosis when sperm and eggs form.
No, and we're not talking about bacteria chopping up viral DNA with restriction enzymes either, because that's attacking foreign DNA, not their own.
Got it. Self-editing.
Exactly. And one more distinction, we're also not talking about the programmed loss of entire chromosomes from body cells, like you see in some insects.
Ah, okay. Why not?
Because those processes seem to be more about variations on how chromosomes segregate during cell division rather than the sort of DNA damage and repair mechanisms we're focusing on today. Our deep dive is really into those deliberate, targeted DNA sequence tweaks.
So how do they actually do it? What's in the toolkit?
Well, the core idea, and this is pretty neat, is that EGE systems often use specialized molecular machinery to damage DNA, but in a very controlled way.
Damage it on purpose.
Yeah. And then they cleverly subvert or redirect the cell's normal DNA repair pathways to rearrange and fix that damage according to the plan.
Okay, so it's like a construction crew that usually fixes broken pipes, but now they're intentionally denting a pipe to reshape it for a specific new purpose and then meticulously fixing their own induced damage.
That's a great analogy, exactly. When DNA gets broken, a double-strand break, or DSB cells have sophisticated ways to fix it, EGE systems often hijack these.
Like what ways?
Well, for instance, if a DSB needs a new protective end cap, like a telomere on a chromosome, they use an enzyme called telomerase. It adds these repeating DNA sequences.
Okay, and what about just sticking broken ends together?
For that, cells often use a quick fix method called non-homologous end joining, NHEJ. It's a bit like roughly gluing two broken pieces together.
Thinking dirty, maybe.
Sometimes, yeah. There's also a slightly more error-prone version called microhomology-mediated end joining, MMEJ, which looks for tiny matching sequences to kind of guide the repair.
And for more precise fixes, if you need to replace missing info.
Then they often turn to homologous repair, or HR. The most common type is called synthesis-dependent strand annealing, SDSA.
SDSA. Okay, how does that work?
Think of it like having a perfect spare part or template somewhere else in the genome. If there's a break, the cell machinery finds an identical, intact DNA segment, uses it as a template to copy the missing information, and then accurately inserts it back into the break. It's incredibly precise.
Wow. Okay, let's pause here a sec. So genome editing isn't just a lab trick. It's happening naturally, all the time, in all sorts of organisms. And the really fascinating part is how they're taking the very tools cells use for repair, fixing broken DNA, correcting errors, and repurposing them for program changes.
It's like seeing that mechanic use a wrench designed for tightening bolts to instead very deliberately shape a new part.
Yeah.
And what's really striking is how these ancient fundamental DNA repair pathways, which normally just maintain the status quo, maintain genomic integrity, are leveraged and subtly tweaked for specific adaptive outcomes.
It's not about inventing whole new toolkits from scratch.
Not usually, no. It's about cleverly repurposing what's already there. So what strikes you most about this initial idea, the cellular retooling? Can you already see the potential for incredible versatility here?
Absolutely. Okay, let's dive into the first major category then, deletion editing.
Right.
This is exactly what it sounds like. Organisms deliberately cut out specific DNA sequences, but crucially, only from their somatic cells, their body cells.
While retaining those sequences in the germline cells, the ones passed to the next generation.
Exactly. It's like having the master architectural blueprint that stays complete, but for the actual house construction, certain non-essential rooms are just, poof, removed from the working copies.
And we see examples of this across the tree of life, sometimes on a massive scale. At the simplest level, there's something called programmed DNA elimination, or PDE.
PDE, okay. Where do we see that?
In nematodes, in tiny planktonic crustaceans called copepods, and even in hagfish. They just uniformly delete large chunks of DNA, sometimes hundreds of genes, sometimes up to 90% of the germline genome from almost every somatic cell.
90%.
Yeah.
So if a nematode has this huge germline genome, its body cells might be running with a much smaller, maybe more efficient version.
Exactly.
Right.
It's incredibly efficient. And then you have single-celled organisms called ciliates. They have 2 nuclei, and they go even further.
Auto.
They actually fragment their somatic nucleus genome into tiny little chromosomes, some as small as half a kilobase. And some species even assemble functional genes in the soma from segments that were scattered around in the germline.
Whoa. Okay, that's like building a complex Lego set, where the instructions first tell you to throw out most of the pieces, then build something functional by assembling the remaining ones from a totally jumbled pile.
It's pretty wild. And then there's VDJ recombination, a really critical system in our own immune system and most vertebrates.
Right, antibodies. This is also deletion.
It is fundamentally a deletion process, yes, but with a very different goal. It generates near infinite variation for our antibodies, immunoglobulins.
How does it do that by deleting?
Well, it takes dozens of available gene segments, randomly picks just 2 or 3, cuts out everything in between, and then ligates those chosen segments together, often imperfectly, into a single functional antibody gene.
Imperfectly, on purpose.
Yes. That imperfection adds even more diversity. It allows our immune system to recognize pathogens it's never encountered before. The potential diversity is staggering. Estimates range from 10 million to maybe 100 quadrillion unique antibody molecules.
That's an incomprehensible number from just deleting and patching.
Pretty much. And it's not just VDJ. There's also class switch recombination, another VDJ-like deletion system that helps diversify antibodies further.
Okay, so how does this deletion mechanism actually work, conceptually?
Typically, it starts when an enzyme, an endonuclease, makes precise DNA breaks, those DSBs.
Not random breaks.
No, they happen at specific developmental times and often target short, conserved DNA sequences like signposts. For VDJ, the RAG1, RAG2 enzyme complex makes these specific cuts.
Then what?
Once the breaks are made, the cell has ways to distinguish which fragments to keep and which to discard. Then the broken ends of the keat fragments are either stitched back together.
Using that NHEJ pathway.
Often, yes. And in BDJ, it's a version of NHEJ that actually introduces small diversity-generating errors, little insertions or deletions at the junctions.
Adding more variety.
Exactly. Or, in other cases, like in those nematodes, the broken ends are simply capped off with new telomeres.
It's really remarkable how these different deletion systems, nematodes versus our immune system, often seem to target similar types of DNA.
That's true. In nematodes, copepods, and ciliates, the deletions are heavily biased toward repetitive sequences, especially those mobile elements, transposable elements.
Like clearing out the junk DNA or potentially disruptive DNA from the active somatic genome.
That seems to be a major driver, yes. And another common feature is that the exact boundaries of the deletions can be a bit variable.
Why is that?
It's partly because NHEJ, the repair pathway, is inherently a bit imprecise. It doesn't always join perfectly, so you see this variability in Ascaris nematodes and some ciliate edits. It might be due to slight variations in where the initial cut happens or how much the DNA ends get trimmed back before joining.
Okay, so why do organisms go through all this dramatic cutting and pasting? The functions seem really diverse.
They are. Genome defense is a big one, especially for those organisms like crustaceans that get rid of loads of transposable elements, protecting the working genome.
Makes sense. What else?
For nematodes, it might be primarily about remodeling telomeres, since the chromosome ends are almost always involved in the deletions.
Okay.
Ciliates, which have these huge cells and need massive amounts of DNA replication for their polyploidy. Well, deletions help simplify that whole process.
Streamlining.
Exactly. And of course, in us jawed vertebrates, the deletions in VDJ are absolutely critical for generating that immense antibody diversity. Our immune system relies on it.
So, connecting back to the big picture, deletion editing really shows this incredible spectrum of strategies for different biological needs.
Absolutely. From drastically shrinking a genome to manage cell processes to generating an almost infinite immune repertoire, organisms get really creative with these cut-and-paste mechanisms. It's all about tailoring the genetic blueprint for the specific job at hand.
So, thinking about this, what does it mean for you, the listener? Can you imagine other scenarios where an organism might benefit from just shedding large chunks of its DNA? What kind of trade-offs do you think might be involved in such, well, dramatic genome rearrangements?
Good questions to ponder.
Okay, moving on to our second major category, insertion editing.
Right. So instead of cutting things out, this is where organisms replace or expand a specific DNA location with a new DNA sequence, adding or swapping information.
And this one has a really interesting dual role, doesn't it? Immune defense versus pathogenicity.
It really does. It's fascinating. Many pathogens actually use insertion editing to basically change their disguise. They replace a gene encoding a protein, an antigen that the host immune system recognizes, by inserting a different version. This lets them temporarily evade detection. It's a constant game of cat and mouse.
So things like the bacteria causing gonorrhea or syphilis or Lyme disease.
Yeah.
And that parasite for sleeping sickness, trypanosoma, they use this.
Exactly. It makes a lot of sense that this ability probably evolved in response to pressure from the host's immune system constantly trying to recognize and clear them.
But then the flip side is that hosts use it for their immune defense too.
Precisely. It's analogous to VDJ in some ways. While humans rely heavily on VDJ deletion for antibody diversity, other animals, like birds and many mammals, generate less diversity that way. Instead, they use a process called gene conversion in cysts.
Gene conversion. Okay, what's that?
It essentially replaces parts of an already assembled immune gene by copying sequences from elsewhere in the genome, from those silent donor loci we'll talk about. It swaps out segments.
I see. And even jawless vertebrates like lampreys and hagfish, which don't have our kind of antibodies.
Right, they diversify their own immune receptor genes. Variable lymphocyte receptors, or VLRs, which function like antibodies using a very similar gene conversion mechanism.
So attack or defense, the underlying strategy often involves this insertion or replacement, and they share a common genomic setup.
Generally, yeah. You typically have an acceptor locus, that's the active gene being edited, and then elsewhere in the genome you have donor loci.
Donor loci.
These can be anywhere from just a couple to over 1,000 copies of related genetic material. They might be intact genes or fragments or non-functional pseudogenes, but crucially, they're transcriptionally silent. They're not being actively used.
Like a library of spare parts.
Exactly. And the editing outcome is usually unidirectional and typically error-free. The donor loci themselves aren't changed. The acceptor locus gets replaced, maybe entirely or just partially, by a sequence copied from a donor or sometimes a mix of donors. And importantly, it retains its protein coding ability and stays active.
It's a very clean copy-paste operation then. Precision work.
Very much so. And the mechanisms, as you hinted, primarily rely on that homologous repair pathway, often SDSA-like processes, using the homology between the acceptor and donor loci.
We have a good example of this, right? Yeast mating type switching.
Yes, that's a classic, well-understood example. Yeast uses insertion editing to switch its mating type, which it needs for sexual reproduction. It starts with a precise DNA break, a DSB, right in the active mating type gene. Then it uses the homology between that broken acceptor locus and the two silent donor loci, which specify the alternate mating types, to guide repair via SDSA, that gene conversion process. A strand from the break invades a donor, copies the silent allele, and then that new copy replaces the original sequence at the active locus.
That's super clever, copying the silent information into the active slot.
And you see variations, like in those... donor gene segments, creating a chimeric or hybrid gene by copying bits from different donors.
Wow, template switching.
Right. And for most other insertion systems, even if we don't know the exact molecular details, the whole setup silent donor arrays, the need for homology, the precise insertion of chimeric genes while keeping the protein code intact, it all strongly points towards these SDSA-like homologous repair pathways being central.
But there are exceptions, right? Especially in bacteria and archaea.
Yes. Major exceptions, particularly in prokaryotes. They often use entirely different mechanisms that don't rely on homologous DNA repair, and these systems tend to expand the acceptor loci rather than just replacing them.
And the most famous example here is CRISPR, isn't it?
Absolutely. CRISPR spacer acquisition. We know CRISPR machinery chops up foreign DNA for bacterial immune defense. But the key part for insertion editing is that it also inserts short DNA sequences from those invaders, the spacers, into the CRISPR locus in the bacterium's own genome.
So it's literally grabbing a piece of the enemy's DNA.
And integrating it into its own genome as a genetic memory, a molecular mugshot.
How does it do the insertion?
It uses a really unique enzyme complex, Cas1, Cas2, that catalyzes a specific Nikken ligation reaction. It's not like standard repair. It precisely integrates these new spacer sequences next to existing ones.
Amazing. And there are other non-homology-based systems in prokaryotes, too.
Oh, yes. Site-specific recombinases are widespread. These are enzymes like molecular scissors and glue that recognize specific short DNA sequences at two different locations. They bind, bring the sites together, and then catalyze recombination cutting and pasting between those sites.
Like what?
A key example is integrons. They use site-specific recombination to reversibly insert silent gene-bearing cassettes, often from plasmids, into an active expression site.
And this has been important for things like antibiotic-resistant spreading.
Hugely important. But they can also mediate other phenotypic changes. And what's really versatile is that very similar site-specific recombinase systems can also cause inversions, flipping a DNA segment around, or deletions, not just insertions. Pathogenic Salmonella bacteria use a site-specific recombinase to flip a DNA segment containing promoter sequences. This switches which flagellar antigen gene gets expressed, effectively changing the bacterium's coat to evade the immune system.
So, insertion editing. It gets really interesting here. It's not just about adding genetic material. It's clearly a strategic move. Evading immune systems or diversifying your own.
Exactly. And the cleverness lies in how organisms either leverage that precise homologous repair copying mechanism or deploy entirely different enzyme systems like CRISPR or site-specific recombinases to integrate new information or switch things around.
This really makes you wonder. Considering the high stakes in these host-pathogen conflicts, why might some organisms rely on potentially error-prone mechanisms for diversity, while others seem to need near-perfect copying? What are the evolutionary pressures driving these different approaches to adding genetic information?
That's a central question in the field.
Okay, let's tackle our final category, substitution editing.
Right. This is where an organism makes targeted changes to individual DNA letters, base substitutions, replacing one base for another, but at rates way, way higher than normal spontaneous mutations, like at least 1,000 times higher.
1,000-fold higher. Okay, so the big challenge here must be control, right? How do these systems hit the right spots and avoid messing up the rest of the genome, especially if they aren't always guided by homology?
That's the key issue. And the mechanisms involved are actually quite diverse, less unified than maybe the other categories.
Okay, like what?
Well, one really well-known example takes us back to the immune system. Somatic hypermutation, or SHM, of immunoglobulin genes in vertebrates.
Another layer of antibody diversification.
Exactly. It happens after VDJ recombination. It starts when an enzyme called AID activation-induced cytidine deaminase initiates the process.
AID. Okay, what does it do?
It chemically modifies cytosine C bases, changing them into uracil U, which isn't normally found in DNA. Now, AID has a specific requirement. They can only really process single-stranded DNA.
Single-stranded. How does that target it?
Because DNA temporarily becomes single-stranded when a gene is being actively transcribed into RNA. So this naturally targets AID activity primarily to genes that are being heavily used, like the antibody genes that have just been assembled by VDJ.
That's clever targeting. So it hits the active antibody genes. Then what happens to that U in the DNA?
That U creates a mismatch, and this triggers DNA repair pathways. But here's the twist. The repair process itself is subverted. Instead of accurately fixing the U back to a C, the cell uses error-prone DNA polymerases.
Error-prone. They make mistakes on purpose.
Effectively, yes, in this context. These polymerases introduce additional substitutions, not just at the original U site, but nearby as well. This process, combined with other ways AID activity is controlled, leads to a shower of mutations specifically within the antibody genes, fine-tuning them to bind invaders better.
Error-prone repair sounds risky.
It is. There's a significant downside. Off-target edits do happen. AID can sometimes hit other genes, and these unintended mutations are implicated in the development of many human cancers. It's a powerful system, crucial for immunity, but it comes with a calculated risk.
Wow. Okay. What's another example of substitution editing?
A really different one is repeat-induced point mutation, or RIP, found in fungi, like the common bread mold Neurospora.
RIP. What does that do?
This system massively mutates almost any duplicated sequence in the fungal germline genome, any sequence that appears more than once.
Any duplication. How specific is that?
It's quite indiscriminate towards duplications. It can target identical repeats as small as maybe 155 base pairs, or slightly inexact ones around 500 base pairs. The thinking is that it's a defense against repetitive elements like transposons, but it hits any duplication.
How does it mutate them? Also AID-like.
It also depends on cytosine deamination, but the mechanism seems different. First, cytosines within the duplicated regions get packed with a methyl group. Then, an unknown enzyme specifically deaminates these methylated cytosines, converting them directly into thymidines, T.
So a direct C to T change, targeted at methylated Cs and repeats.
Exactly. And importantly, unlike SHM, there's no further diversification step here involving error-prone DNA repair. It's just that specific C to T conversion.
But does it stay perfectly targeted to the repeats?
Not entirely. RIP frequently leaks. It causes C to T substitutions in unique sequences next to the targeted duplications. So it's a powerful defense, but maybe a bit messy, potentially hitting nearby genes. It seems there might be some kind of direct DNA alignment guiding it, but it's not perfect.
Okay, one more. You mentioned DGRs.
Right, diversity-generating retroelements, or DGRs. These are found in prokaryotes, bacteria, and archaea.
How do these work? How do they target?
Here, the locus specificity is basically built right into the editing machinery itself. It's usually encoded as a small cluster of adjacent genes. This includes the target gene, called the focal gene, which has the sequence to be edited. Right next to it is the gene for an error-prone reverse transcriptase enzyme and an unedited donor sequence that serves as a template.
A reverse transcriptase, so RNA is involved.
Yes. When the donor sequence is transcribed into RNA, the tail end of that RNA molecule does something amazing. It binds to and specifically activates the reverse transcriptase.
Okay.
Now, this reverse transcriptase makes a DNA copy, cDNA, of the RNA template, but it's deliberately error-prone in a very specific way. Wherever the RNA template has an adenine A, the reverse transcriptase incorporates an arbitrary nucleotide into the new DNA strand.
Arbitrary, like it just picks randomly between A, C, G, or T at those spots.
That's the current understanding. It introduces massive targeted variation at specific positions corresponding to adenines in the template RNA.
That is wild. What happens to that mutated cDNA?
Guided by sequence motifs, this highly edited cDNA then somehow replaces the original sequence in the focal gene. The exact recombination mechanism isn't fully understood, but it's not standard homologous repair. The result is base substitutions targeted very specifically to certain nucleotides within that focal gene at an incredibly high rate, maybe 40% or more at the targeted position.
40%. Okay, so substitution editing. The mechanisms are all over the place, but the outcome is always these super high mutation rates at specific spots.
Exactly. Whether it's fine-tuning an antibody with SHM, silencing duplicated sequences with RIP, or rapidly changing surface proteins with DGRs for survival, maybe immune evasion, or signaling these systems show remarkable ingenuity in rewriting single letters of the genetic code.
So thinking about this, especially the potential for off-target edits in something like SHM, which is vital for our own health, what does this mean for you listening? How might understanding these natural mechanisms help us improve our own gene editing biotechnologies, like CRISPR? Can we learn to be as precise, or maybe even more precise, than nature?
It's definitely a major goal of the field.
So what started as this kind of surprising idea, organisms intentionally changing their own DNA, it really unfolds into this grand story of ingenious biological engineering.
It really does. From tiny nematodes to our own complex immune systems, life finds these incredibly diverse ways to dynamically rewrite its own blueprint.
Yeah. We've looked at deletions, insertions, and substitutions. And across these categories that seem pretty different on the surface, some common themes start to emerge, don't they?
They really do. Like, how many of these editing systems use specialized machinery to damage DNA in a controlled way?
Making breaks or mismatches.
And then they subvert the cell's normal DNA repair pathways to fix that damage in a way that results in an edit, often guided by sequence motifs or homology. Repurposing is key.
With some exceptions, like those site-specific recombinases.
True, those are different. Another big theme is function. So often, editing seems tied to conflict.
Host-pathogen conflicts, yeah. Diversifying immune receptors in the host, or letting pathogens switch their antigens to hide. It's like a biological arms race played out in the genome.
Exactly. Or it's about genome defense, inactivating or deleting those mobile genetic elements, the transposons, that can cause trouble.
And the proteins involved. Even in organisms that are incredibly far apart evolutionarily, the actual molecular tools can be remarkably similar.
That's striking. Many of the endonucleases that make the initial breaks for insertions or deletions seem to be derived from transposases, the enzymes that help transposons jump around.
Ah, so the tools for making unwanted jumps are repurposed for controlled editing.
It looks that way often. And those site-specific recombisases we mentioned, also related to transposases, and deaminases like AID, turn out to be incredibly versatile tools for different kinds of editing, generating mismatches or even initiating breaks.
Which leads to maybe the most startling observation, this idea of convergent evolution.
Yes. Highly similar editing systems seem to pop up again and again in very distantly related organisms, suggesting they evolved independently.
Like how jawed vertebrates, like us, and jawless vertebrates, like lampreys, came up with different genes and slightly different mechanisms, but ended up with functionally similar immune systems based on programmed gene rearrangement.
Right, or how bacteria, single-celled eukaryotes like Trypanosoma, and even fungi independently evolved antigen-switching systems that use similar homology-directed insertion mechanisms, even though the actual genes being switched are totally different.
Even yeast mating type switching shows convergence between distantly related yeast species using different ways to make the initial DNA break.
So why does this happen? It strongly suggests that organisms have this sort of fundamental toolkit already available, things like transpose-derived enzymes, deaminases, the basic DNA repair pathways. And from this common toolkit, diverse and complex editing mechanisms can apparently be assembled multiple times when the evolutionary pressure is strong enough, like facing down pathogens. Maybe creating a new editing system isn't as evolutionarily difficult as we might think if the parts are already lying around.
That's a fascinating perspective. But you mentioned earlier that despite how important EGE is and how much sequencing we do now, the discovery of brand new systems has kind of slowed down.
It has, surprisingly. And it's probably not because there are no more out there to find.
So why the slowdown?
Well, partly it's a focus issue. Research tends to concentrate on well-studied model organisms and human pathogens, so we might be missing things in less studied branches of life.
Okay.
Also, some older techniques, like looking carefully at chromosomes under a microscope, were actually quite good at spotting large-scale elimination events that might be missed by standard sequencing analysis pipelines. And there are technical hurdles, too. Edits might only happen in specific tissues or at specific times. The DNA regions involved often contain highly repetitive sequences or arrays of very similar genes, which are notoriously difficult to sequence accurately and assemble correctly using standard short-read sequencing. Reads might map ambiguously.
But these challenges aren't impossible to overcome, right?
No, not at all. It requires different approaches. Using PCR-free long-read sequencing technologies can help span those complex regions. Careful bioinformatics analysis looking specifically for rare sequence variants or unusual patterns at highly variable loci is key. And ultra-accurate sequencing methods can help distinguish true low-level editing from sequencing errors.
So where should we be looking for these undiscovered systems?
Most likely in overlooked organisms. It's striking that there's still no confirmed endogenous editing system known in the nuclear genomes of plants, for example. We should look for telltale signs. Genomes with large arrays of similar genes or gene fragments, or organisms with unusually large or small genomes, which might hint at genome defense mechanisms being active.
But the biggest clue might be function.
I think so. Given how often editing systems arise in the context of immune conflicts, the most promising places to find new editing systems and maybe even entirely new mechanisms are likely within understudied pathogens and their hosts. That ongoing evolutionary battle is a powerful engine for genetic innovation.
We've really only scratched the surface here today, haven't we? It makes you think. If our understanding of even our own genome is constantly changing, what incredible secrets are still locked away in the genomes of all those less studied organisms out there, especially the ones locked in these constant battles?
Absolutely. This whole journey through deletions, insertions, and substitutions, it reveals this profound theme in evolution. Innovation often comes not from inventing something totally new, but from repurposing what's already there. And that leaves us with a big question for you to think about. If these complex editing systems can arise convergently, again and again, from this common biological toolkit, what other hidden genetic capabilities might be waiting to be discovered, especially in those vast, unexplored corners of the tree of life?
And what new biotechnologies might emerge once we understand these deep, fundamental processes better?
Exactly. The next great breakthrough in genetics might just be waiting in the most unexpected place.